So here's an example of what's called dominant epistasis, where a dominant allele at one locus masks an allele at the second locus. And so they give an example here of dominant epistasis with squash fruit color, where if the squash has an A allele, the A allele is dominant. So if squash has an A allele, it will be white no matter what. Even it doesn't matter what the B allele looks like. So we can do this problem as well. I'm just writing the notes down on my iPad, and then we'll transfer over to the iPad. Okay, so let's go to the iPad. So the way it's phrased on the slide is a little bit confusing, but basically what we're looking at is an individual that has a big A allele will always be white. So A controls deposition of white. And then B controls the other color. So we can do the same problem here. We get that 3/4 white and 1/4 other color. And the other colors are yellow and green. And so again, this is just heterozygote, a classic Mendelian type of dominant recessive relationship. So the heterozygotes being crossed will give us that 3/4 ratio. So 3/4 will have the yellow color. Big B basically gives yellow color, little b gives green color. 1/4 green, 3/4 yellow, 1/4 green. And now if we do our crosses, 3/4 times 3/4 is 9/16, and because they are white, it doesn't matter what's here, these are white. 3/4 times 1/4 is another 3/16, and these are also white. 1/4 times 3/4 is 3/16 that actually can express their normal color. So these are going to be yellow. And 1/4 times 1/4 is 1/16 can be green. So again, we have to combine these because they're the same phenotype. So we have 12 out of 16 that are white, 3/16 yellow, 1/16 green. So our final phenotype ratio is 12 to 16 to 1. And so that's, you see how if the allele is recessive, it gave us the different ratio we saw with the mice, that like 9 to 1 ratio. But if the epistasis is dominant, we got that 12 to 3 to 1 ratio. And we also have situations where you could get almost like a blended or a novel phenotype. And so in this case, we're also talking about squash. And so here we have squash shape. And so if a fruit is dominant, right, at both of those loci, the big A, big B, that is a disc-shaped fruit. If it is homozygous recessive, that is an elongated fruit. And when you cross them together, like you might expect, when you get the heterozygote is also disc-shaped. Now, what happens is when we cross the heterozygotes and we start to get blended genotypes where one allele pair, or, like one gene pair has a dominant, but the other one's recessive. So I'm going to transfer over to my screen so we can take a look at that. Okay, so in some way, both of these genes, both A and B, right, are responsible for the disc shape. And little a and little b are both responsible for the long shape. So as long as you have one big A and one big B, you will get a disc, like right here. If you have all little a's and all little b's, your fruit will be long. But what happens if you have, right? Well, this would obviously be, right? So what if you have the dominant A phenotype with the recessive? So you basically have disc here, but it says long here, or you have the opposite, where you have long here and disc here. So classically, when we think of something like this happening, right, we think that what we should get is this fruit should be disc, and so should this. That's what we think. So we can set up our cross. So 3/4 big A, 1/4 little a, or we can just say, we can even do it like this. We'll just use the phenotype because that's what we've been doing the whole time. So 3/4 we would expect to have the disc, 1/4 long from the A allele, and then we also have the B allele, which kind of does the same thing. 3/4 disc, 1/4 long for both. Sorry, I have a rooster and it's like 5:30 in the morning, so he's up. Okay, and again we get that 9, 3, 3, 1 ratio. So here I'm going to consider both gene pairs, right? So I have disc, disc. So that is definitely going to be a disc. Here I have disc for A, but long for B. What is that going to be? Here I have the opposite. I have long for A and disc for B. And then here I have long for A and long for B, which I know is going to be long. So these two that I don't know, what it turns out is that when you have only one gene pair that has a heterozygous or that has the dominant allele and the other gene pair is recessive, you actually do get a blended phenotype. And so these fruit end up being sphere-shaped. And so you actually end up with a 9:6, because you have to add up the two spheres, to 1 ratio of disc to sphere to long. So, if I gave you a problem like this on your first exam on Tuesday, that problem, I would definitely define that relationship for you. I wouldn't leave you hanging and be like, okay, well, you have to know that if it's a disc and a long, you get a sphere. I would tell you that if you have, one allele or one gene pair where both alleles are recessive and the other gene pair one of the alleles is dominant and you get this intermediate shape, I would tell you that. I would just expect you to be able to do the math to show me why. So let's just wrap up this section. And this will be the last material that we'll put on your first exam. So again, the idea is that we just looked at, if you have a disc-shaped fruit, that requires that you have a dominant allele for both loci. Genes A and B have to have a dominant allele. To get a sphere-shaped fruit, you have to have dominant at one allele or the other. It doesn't matter, but not both. You have to have it at one only. And if you have no dominant alleles, then you get the long fruit. And so this is what these look like. So this is the disc-shaped fruit. This is a spherical fruit, and this is a long one. So now you can think of like your zucchini and squash. They are recessive, homozygous recessive for both genes controlling the shape of the fruit. And we also have these types of gene interactions that can occur in things like Drosophila as well. And this is controlling the eye color and the mixing of pigments. So each step of the pathway has its own enzyme, and so you have separate genes that end up controlling the eye color. And so here they're showing you there's one set of genes that'll give you red, one set that gives you brown. If they are both functional, the eyes are red. If the red is not functional, then the eyes would be brown. So it doesn't really matter what the brown is doing as long as the red is functional. And then here, if red is functional and no brown, the eyes are still red. And they're kind of like different shades of red a little bit, but that's okay. They're still red. And then of course, if both of the genes are mutated, you have no color in the eyes. So these types of interactions, you can still do, your forked diagrams, you can still do your Punnett squares a lot of the time, but you just have to be cognizant of and thinking about what those ratios will end up looking like.